Have you ever looked closely at how we age? Some people seem to maintain vibrant, youthful energy well into their late 70s, while others seem to experience sudden structural decline almost overnight. Why is that? Is aging just a random, chaotic breakdown? Or is it governed by a hidden biological script operating beneath our awareness? Welcome back, friends. Today, we are diving into a profound biological mystery that sounds almost like science fiction, but is deeply grounded in modern biogerontology. It turns out that your cells are constantly participating in a high stakes tug of war between two opposing chemical signals. In the world of longevity research and clinical physiology, scientists often refer to this balance using a simple shorthand: the C6 and C10 pathways. Think of it as a cosmic dualism written directly into your biology. One side represents systemic decay and demolition, while the other coordinates regeneration, energy production, and survival. When these two forces are in harmony during youth, your body heals rapidly. But as the decades pass, a silent shift occurs, tipping the scales toward a permanent state of structural breakdown. To understand how to alter this trajectory, we first have to unmask the hidden villain of the story, a phenomenon researchers call the C6 drip. Imagine waking up every single day to an unending internal winter. Your joints feel stiff, your energy levels fluctuate, and your muscles gradually feel less stable. This isn't just a consequence of getting older, it is driven by a specific immunological breakdown. When we lead a physically stagnant lifestyle, encounter chronic emotional stressors, or consume highly processed diets rich in refined sugars, our fat tissues and immune systems get confused. Instead of protecting us, they begin to release a tiny, constant, background trickle of a pro-inflammatory protein called interleukin 6, or C6. This persistent background leak is known as the C6 drip. Now, you might think a tiny leak wouldn't do much damage. But researchers at institutions like Harvard and the National Institute on Aging have found that this chronic elevation of C6 leads to a destructive state called inflammaging. It acts like a slow biological acid, causing gradual vascular stiffening, synaptic degradation in the brain, and the steady wasting away of skeletal muscle mass, medically referred to as sarcopenia. Studies analyzing older individuals experiencing muscle loss show a direct, negative correlation between elevated background C6 levels and their overall muscle index. It keeps the body in a constant catabolic state, breaking down infrastructure without ever hitting the rebuild button. But wait, if C6 is so destructive, why did nature design our bodies to produce it in the first place? This brings us to a mind-blowing physiological paradox. What if I told you that the key to turning off this chronic, destructive leak is to shock your body with a massive, explosive surge of the exact same chemical? It sounds completely counterintuitive, right? Why would you want more of the molecule causing the decay? Here is where the beauty of human physiology reveals itself. When you engage in vigorous, sweat-inducing physical exercise, your contracting muscle fibers act as a powerful endocrine organ. As your muscles experience mechanical tension and microscopic structural trauma, they suddenly trigger an acute, massive explosion of muscle-derived C6, sometimes increasing blood concentrations up to 100-fold. But here is the critical difference. Unlike the chronic immune trickle born from sitting on the couch, this exercise-induced spike occurs in a completely different environment. Instead of driving destruction, this massive surge acts as a vital metabolic sensor. It tells your liver to mobilize glucose, enhances insulin sensitivity, and signals your body to burn fat. Even more astonishingly, The body's cellular wiring is calibrated so that this high-magnitude spike of acute C6 is the exact key required to unlock the hero of our story, cytokine 10, or C10. Roughly 1 hour after an intense workout, your circulation becomes flooded with C10, scientifically known as interleukin 10. Once released, C10 acts as the ultimate master repair coordinator. It downregulates the inflammatory machinery, switches your tissue macrophages from destructive clearers to pro-regenerative healers, and signals your cells to build brand new mitochondria, the power plants that keep you feeling young. The decay caused by the microtrauma of exercise is what literally forces the body to unlock systemic renewal. Let's zoom even closer into the cell, down to the level of energy production. As our tissues age, they often experience a severe fuel bottleneck. Our cells normally burn long-chain fatty acids for energy, but this process relies heavily on a specialized molecular transport system called the carnitine shuttle. Over time, cumulative mitochondrial damage and diminished transport proteins cause this shuttle system to break down. The result? Cellular traffic jams, metabolic inflexibility, and insulin resistance. But nature left a fascinating backdoor, and it involves another dimension of the C6 and C10 pathways, medium-chain fatty acids. Caproic acid, which contains 6 carbons, and capric acid, which contains 10 carbons, possess a unique chemical structure. Because of their lower molecular weight, research shows they enter the mitochondria completely independently of the broken carnitine shuttle. They simply diffuse directly across the inner membranes to provide an immediate, reliable energy source for demanding organs like your heart and brain. Furthermore, research suggests that capric acid does not just serve as fuel. It acts as a direct genetic signaling ligand. It binds to metabolic receptors like PPR gamma and activates NAD-dependent enzymes, known as sirtuins, specifically SIRT1 and SIRT3. These longevity pathways help stabilize your epigenetics, clear out damaged cellular debris through a cleanup process called autophagy, and significantly reduce the leakage of harmful reactive oxygen species. In the brain, these compounds help generate efficient ketone bodies, providing a metabolic alternative that may support cognitive resilience against age-related decline. The deeper we look, the more we realize that the C6 and C10 labels appear across the entire landscape of longevity science, including our joints and structural matrix. For instance, your joint cartilage relies heavily on a protective mesh known as collagen 6, or C6. This structural shield acts as a mechanical filter, ensuring your cells interpret physical forces correctly to synthesize youth-preserving elements like agrecan. However, when joint tissue degrades through injury or chronological aging, a pathological shift occurs. The protective collagen 6 network breaks down, and chondrocytes begin abnormally producing collagen X, or C10. Instead of protecting the joint, collagen X causes matrix calcification, turning compliant cartilage brittle, rigid, and highly susceptible to shear damage, ultimately driving the progression of painful osteoarthritis. Simultaneously, at the cellular level, scientists are studying how synthetic short-chain ceramides, also labeled C6 and C10, affect cellular fate decisions. In early experimental laboratory stages, these cell-permeable compounds are being studied for their potential to selectively punch holes in the outer membranes of senescent cells, often called zombie cells and malignant tumors, triggering programmed cell death while sparing healthy tissue. While this field of senolytic intervention is incredibly exciting, It is vital to remember that it is currently restricted to early-stage laboratory models and is not available as a human anti-aging therapy. Finally, we must ask, how does aging spread across the body? When cells become senescent, they don't fade away quietly. They begin secreting a toxic inflammatory cocktail called the senescence-associated secretory phenotype, or SASP. Key players in this secretome include powerful chemokines like CCL6 and CXCL10. When CXCL10 accumulates in the system, it initiates a destructive intracellular cascade. It triggers internal calcium surges and recruits the IKK complex, which degrades protective cellular barriers and allows a pro-inflammatory protein called NFKB to translocate directly into the nucleus. Once inside, it activates the transcription of a massive wave of pro-inflammatory genes, amplifying tissue breakdown across the entire organism. We see this nonlinear breakdown mirrored throughout nature. In botanical sciences, when seeds age, they hit a precise, critical node at day 10 where their structural carbohydrates collapse and their cellular envelopes fragment entirely. Even atmospheric aerosols undergo a parallel C6 to C10 photooxidation and decay process. Decay and growth are intertwined rules of the physical world. So, knowing this science, how do we tilt our internal scales away from the chronic decay of the C6 drip and toward the regenerative power of C10? Clinical frameworks like Dr. Henry Lodge's Harry's Rules outline practical, evidence-based behavioral strategies to do exactly that. Exercise 6 days a week. Engage in at least 45 minutes of daily physical exertion, balancing aerobic endurance and strength training. This intentional mechanical stress is what forces the acute muscle-derived C6 spike to unlock the downstream C10 rejuvenation cascade. Avoid processed foods. Eliminate refined sugars and trans fats to lower the baseline systemic inflammation that feeds the chronic background leak. Cultivate deep social connections. Actively foster friendships and community bonds to suppress the nervous system's alarm responses, reducing stress hormones that trigger pro-inflammatory cytokine leaks. Pursue passion and purpose. Dedicate yourself to demanding meaningful goals to promote cognitive resilience and neuroprotection. Before you implement any changes, please remember that the scientific research we discussed today is strictly for informational and educational purposes. It does not constitute medical advice, diagnosis, or treatment. Always consult a qualified healthcare professional before modifying your physical exercise routine, dietary patterns, or supplement protocols. While research suggests these interventions may support longevity pathways, they are not guaranteed to prevent, cure, or treat any medical condition. Isn't it fascinating how our biology is wired to require controlled, acute challenges in order to activate its deep, restorative power? What part of the C6 and C10 balance surprised you the most? Let me know your thoughts in the comments below. Make sure to hit that subscribe button, and let's continue decoding the mysteries of the human body together. Stay curious, and see you next time.